Hello, this is Project 2 Antigen Capture, Presentation, and Recognition in Adaptive Immunity. It's by Kylie Hernandez, Rowan Randolph, and Jorge Escobar. What is an antigen? Antigen is any substance that is recognized by lymphocytes or antibodies as foreign, usually triggering an immune response. Epitope, also known as an antigen determinant site, are sites on or in antigens with which antibodies and T receptors react. As you can see here in this image, the epitope or the antigenic determinant, it's interacting with the antibody and the antigen as shown. B versus T lymphocytes. TCRs recognize peptide fragments bound to MHC molecules, so T cell mediated responses occur only against protein antigens that are synthesized within or taken up by host cells and processed for MHC presentation. BCRs recognize intact native antigens without requiring processing or MHC presentation. Another way they differ is they can bind proteins, polysaccharides, lipids, nucleic acids, either in soluble form or on cell surfaces, typically recognizing their native structure as well. As you can see here in this image, the antigen and the antibodies interacting with those antigens. And as you can see here in this image, how the T cell is interacting with the MHC molecule. MHC molecules and antigen presentation. So for the MHC classes, class 1 has gene products found on unnucleated cells and participate in antigen presentation to CD8 lymphocytes. They're able to eliminate virus-infected and tumor cells. For class 2 molecules are found primarily on antigen-presenting cells such as macrophages and dendritic cells, and participate in antigen presentation to CD4 lymphocytes, or T helper cells, which is able to activate macrophages and help B cells produce antibodies. And as can be shown in the image, the MHC class 1 and class 2, and as you can see, MHC class 1 has one chain compared to class 2. And for antigen processing pathways, class 1 has the endogenous pathway and class 2 has the exogenous pathway. For dendritic cells are professional antigen presenting cells. They are able to capture antigens in peripheral tissues and migrate to lymph nodes, as can be seen in the image, and they are able to process antigens and present them on MHC 1 and 2, and provide co-stimulation to activate naive T cells, and they help bridge innate and adaptive immunity. For cross-presentation, it allows for extracellular or cell-associated microbial antigens to trigger CD8 T cell responses to MHC 1. And dendritic cells ingest those infected cells or microbial agents, which allows for that cross-presentation.
Okay, so BCR and TCR structures. Now, these two are a little bit different. For the B cell receptor, you have the FAB and FC region. You have 2 light chains and 2 heavy chains. You also have the hinge region, which is right about like right here. And all these are linked by disulfide bridges. And for the T cell receptor, you have 1 alpha and 1 beta chain, so only two chains, 1 constant and 1 variable region, and they're also linked by disulfide bonds. And in order to recognize these antigens, the BCR is going to recognize it from these two variable regions, and the TCR is going to recognize the antigen through this one region right here. Diversity of antigen receptors. So this process typically happens before the lymphocytes mature, usually in the thymus or the bone marrow. And what happens is that these antibodies, their genes start to they go through recombination, and what happens is that they kind of scramble into to different sorts of DNA. So it goes through like transcription, RNA processing, translation, and eventually you get millions of different types of antibodies that can detect many types of antigens. Affinity and affinity maturation. So affinity is the binding strength of an antibody. So basically it's going to be like it quantifies the strength between the antibody and the antigen, while affinity maturation is whenever the binding strength increases, which typically happens after the first immune response. So that way the second immune response is much stronger.
Avidity is the total strength of an antibody-antigen binding interaction. So it's essentially a sum of an antibody's affinities. Lymphocyte development of all leukocytes begins with hematopoietic stem cells in the bone marrow. Different signals will activate different lineage-specific transcription factors that determine if the lymphoid progenitors will continue maturation in the bone marrow to become B lymphocytes or migrate to the thymus to mature into T lymphocytes, both having to go through a series of checkpoints along the maturation process. These checkpoints are where T and B lymphocytes will undergo positive and negative selection. Positive selection ensures that T lymphocytes are MHC restricted and that B lymphocytes express a functional BCR, while negative selection basically ensures self-tolerance. If either BCRs or TCRs are too reactive to self-antigens, then they are eliminated by apoptosis. This image is just showing the process by which T and B lymphocytes undergo maturation. It shows how pro T cells travel from the bone marrow to the thymus to continue maturation. And from there, pro T cells must form a TCR beta chain. And at this point, they are double negative, having neither CD4 or CD8 co-receptors. And they must have this beta chain in order to become pre-T cells, while pro B cells must express a functional immunoglobulin heavy chain in order to become pre-B cells. From there, pre-B cells and T cells go through another checkpoint, this time ensuring that pre-B cells have both a light and heavy chain, and that pre-T cells express an alpha chain to complete the TCR and become a double positive T cell now, which means they express both CD4 and CD8 co-receptors. From this checkpoint, pre-T and B cells are able to go undergo gene editing, which is sort of their last chance at survival in case they are self-reactive at this point. And in antibodies, the light chain is what will be edited in order to give it a second chance and be chosen to survive and become a mature, naive B cell. This image is also just showing positive and negative selection of T cells. It shows how the MHC class 1 and MHC class 2 are chosen. If there is a weak recognition of MHC class 2, then these double positive immature T cells are chosen to become mature CD4 T cells. If there's a weak recognition of MHC class 1, then these double positive T cells are then chosen to become mature CD8 T cells. But if there is no recognition of the MHC peptide, then these are negatively selected for apoptosis, as well as if there is a strong recognition of the MHC class 1 or class 2 N peptide. These will also be negatively selected for apoptosis, as we don't want extremely reactive, self-reactive T cells because that will result in no self-tolerance. These are just the rules of our group. These are some of our references, and thank you for listening to our presentation.